Welcome to my channel Pharma Knowledge Forum. In this session, I am going to discuss application of perchloric acid in pharmaceutical industries. Perchloric acid titration is widely used in industries, especially in acid test, because it is easy, fast, and cost effective. It gives accurate and precise results. But many professionals have doubt related to its concept, application, and method development. So I decided to share my skill-based knowledge on this topic. Enjoy this session and clear all your doubts related to perchloric acid titration. In this lecture, you will learn what is perchloric acid titration, why is it needed, why glacial acetic acid is used, why is acetic anhydride added while preparing 0.1 normal perchloric acid volumetric solution, how is normality of 0.1 normal perchloric acid estimated, which type of samples are analyzed by perchloric acid titration, why mercuric acetate is used while titrating halogen acid drug substances salt? What precautions should be taken while performing perchloric acid titration? Which indicator is used for non-aqueous titration? What is the shelf life of perchloric acid? Perchloric acid titration. Perchloric acid titration is a non-aqueous titration. Now question is what is non-aqueous titration? In non-aqueous titration, the analyzed substance is dissolved in a solvent which does not contain water. It means solvent must have zero water. Perchloric acid is used as a titrant. Perchloric acid is strongest acid. Glacial acetic acid is used as dissolving solvent. Now question is why glacial acetic acid is used? I am going to explain the same in next slide. Its principle is similar to that of acid-base titration, means analyte is basic in nature and it accepts the proton from perchloric acid and reaction takes place. In this titration, solvent enhances the strength of perchloric acid to get sharper end point. Now question is what is the need of perchloric acid titration? To get answer of the same, first we should understand the limitation of aqueous acid base titration. Water contains both hydronium ion and hydroxide anion. Hydronium ion and hydroxide anion. Due to these hydronium ion and hydroxide anion, water behaves as a weak base and weak acid as well and can compete in proton acceptance or proton donation with other weak acids and bases dissolved in it. Hence, aqueous acid-base titration cannot be used for two weakly basic compounds and two weakly acidic compounds. Secondly, aqueous acid-base titration cannot be used for those substances which are insoluble in water. Now question is, what are advantages of non-aqueous titration? I am going to discuss in next slide. Advantages: many substances that are not soluble in water and easily soluble in glacial acetic acid and hence can be easily titrated with perchloric acid. Weak bases and weak acids can be easily titrated. Non-aqueous solvents are useful for titration of very weak acids or bases that cannot be titrated in water. Non-aqueous titration are easy, precise, and accurate, fast analysis and cost effective. Now question is, what is the procedure of performing perchloric acid titration? Which type of solvents are used as a dissolving medium? Titrant 0.1 normal perchloric acid or perchloric acid of other strength can be used. There are four types of solvent. Protophilic solvent, those solvent which can accept the proton, for example, basic solvent. Photogenic solvent, those solvent which can generate the proton, for example, acidic solvent. Amphiprotic solvent, those solvent which can accept the proton as well as generate the proton. Example we are going to discuss. Aprotic solvent, those solvent which can neither generate the proton nor accept the proton, generally organic molecules like carbon tetrachloride, chloroform. Glacial acetic having both ability to accept the proton as well as donate the proton. That is the reason this solvent is used in perchloric acid titration. Now question is why glacial acetic acid come under amphiprotic solvent? I am going to discuss in next slide. In some cases, mercuric acetate in glacial acetic acid is also used as dissolving solvent. I am going to discuss in next slide in details. Indicator is a crystal violet compound which can be analyzed maybe amines or amine salts, amino acids and salts of acid. Endpoint can be determined by two modes, by color change of the indicator or by potentiometric titration. Now question is why acetic acid behaves as a amphiprotic solvent. In presence of base, acetic acid donate the proton to base with formation of state anion, but in the presence of strong acid, it accepts the proton with formation of onium ion and chlorate anion. Since acetic acid behaves both as a acid as well as a base, that is the reason it has amphiprotic nature. Case study 1. Titration of weakly basic molecules like aniline, atenolol, quetiapine, fumarate. Sample is dissolved in acetic acid and titrated with perchloric acid either using crystal violet indicator or by titrating potentiometrically. Now question is what is the mechanism? For example, aniline. Aniline is a weak base. Acetic acid donate its proton to aniline and anilinium ion is formed with the formation of acetate anion. Aniline content is determined based on acetate anion. Now perchloric acid react with acetic acid with the formation of onium ion and chlorate anion. This onium ion react with this acetate anion with the formation of acetic acid. This is the mechanism. Case study 2. Titration of halogen acid salts of base, for example, aniline hydrochloride, ethambutol hydrochloride, ethidine hydrochloride. Sample is dissolved in acetic acid containing mercuric acetate solution and titrated with perchloric acid volumetric solution either using crystal violet indicator or by titrating potentiometrically. Now question is what is the mechanism why mercuric acetate solution is used? In acetic acid, aniline hydrochloride exists like that. These chloride anion is too weakly basic to react with perchloric acid and hence it cannot be titrated with perchloric acid. because end point cannot be achieved. Mercuric acetate replaces chloride ion with the formation of acetate anion, which is a strong base. This acetate anion also determines the content of aniline hydrochloride. Finally, this anion, that is acetate anion, react with perchloric acid with the formation of acetic acid. This is the mechanism of halogen acid salts of base. Limitation: normality of perchloric acid must be determined prior to titration. Temperature, moisture, CO2 should be controlled. Solvents are expensive. Volatile solvent can pollute environment. Nasty smell. Preparation of 0.1 normal perchloric acid or 0.1 normal acetous perchloric acid. 8.5 ml perchloric acid, which concentration is about 70%, is transferred in a 1000 ml volumetric flask containing 500 ml glacial acetic acid and 25 ml of acetic anhydride. And finally, volume is made up to 1000 ml. Now, question is, why acetic anhydride is used during preparation of 0.1 normal perchloric acid? Acetic anhydride absorbs the water of the acetic acid with the formation of acetic acid by this mechanism. That is the reason acetic anhydride is added during preparation of zero point 0.1 normal perchloric acid. That is all about this session. Please like at least one of your learnings in the comment section. I am on mission to train more and more pharma professionals by sharing my skill-based knowledge. But if you join my channel, then only I can help you. Please subscribe to my channel.